Just take a pause and get ready to understand 3 big pieces of this manufacturing process from the page on your screen. This page on your screen will tell you critical steps of the process or some important ones that are core of the process, and simply how to handle them. Even some outside visitors like scientists and other researchers can guess from these 3 points an overall process of a manufacturing. Let's understand that in a layman's term and at the same time understand some precautions that this page needs to tell you. Let's look at the annotation number 1. The yellow circled instruction is telling you to use a nitrogen blanket whenever possible during reactions or holds. Let's understand what is that. Nitrogen blanket is a term that provides a flow of nitrogen gas into a chamber or compartment to kill the oxygen present in it. So, no contamination happens. This results as no bacteria can survive. We learned this concept in glove box training. That also means that at this step drug could be exposed to the air. That's why we are blowing nitrogen gas on it, and since it says during reactions, nitrogen blanket is needed, it simply means that there would be a time in bioreactor process when chemical reaction would happen. At that time, you must use a nitrogen blanket. This is clear. Look at the annotation number 2. It is talking about a sampling loop, which is required when you take the samples from the reactor or a vessel, which means this is a strict rule first of all, and it also tells you that there should be a tap-like loop attached to the reactor and sample of the drug should be only collected through this loop, not any other way. It also gives a knowledge to a layman how a sample is collected from a reactor, or at least one way of doing it. There is another critical thing here, which is temperature. Instruction is telling you to continuously monitor and record the temperature of the mixture throughout the period and should be between the prescribed range. This confirms our knowledge that drug manufacturing is like cooking, and one critical aspect is the specific temperature range. At the bottom of the page, there is annotation number 3. It is all about handling the waste, or the byproduct, as we call it. EPA, the Environmental Protection Agency, requires you to dispose of these bio-waste under certain guidelines. Your job will be to transfer those chemicals to their designated and marked containers. An EHS department individual will come and take the waste. These instructions can be such as how to handle a spill, how to discard, and many other instructions on handling the waste. Most critical of all would be not to mix certain chemicals. That's all for this page. So far, the real steps of manufacturing has not started yet, but we are all prepared and ready to go. We have talked about line clearance many times from terminology class and day-to-day responsibilities of manufacturing technician. Now this is the place in BPR. Before you start performing any action, quality assurance person will come to the manufacturing floor to ensure all materials are gathered in manufactured area. All equipments are properly cleaned and status tags indicate that they are ready for use, as you can see on the screen in orange circle. After checking all is okay, QA signs off at line clearance space, as you can see it on your screen circled in green. Step 1 again talks about checking all equipments for their clean and ready to use tags before beginning the operations. Step 2 talks about to ensure that a split butterfly valve is installed within the bioreactor. Step 4 instructions are to start the nitrogen blanket and ensure the nitrogen pressure going to the bioreactor is less than 1 bar. Bar is the unit of nitrogen pressure. On the right-hand side, you see 2 columns. One is for the operator that executes the steps and record readings for equipments, weights, and other raw data in operation performed by column, along with date and time. The column on the far right is for technician, who verifies the same activity. Do you remember the dual control? It is an important FDA compliance part, as verifier provides real-time authentication of the operation executed. Throughout the BPR, these 2 columns on the right for operator and verifier must be filled after every step execution. On your screen, you see step 5 is talking about charging the first chemical to the reactor. And in this step, the weight of the naphthol chemical is provided. Also, the material number of chemical naphthol is given. This is to ensure accuracy of the quantity of the chemical and no mistakes. On the Verify By column, you also see that verification of the weight of the chemical material code and lot number must be verified also. This is just to ensure that we are making no mistakes here. If you look at step 6, 7, and 8, these steps are instructing to add next chemicals in sequence to be charged to the reactor one by one. Since you have mixed all chemicals in the bioreactor now, do you remember the principles of cooking and drug manufacturing? Yes, temperature, agitation, pressure, and time, these are the conditions for the chemical reaction. So the first step on this page is instructing the technicians to set the agitator speed to 100 RPM. This agitator is kind of a propeller inside the reactor to keep mixing the chemical. This step also instructs to keep the agitation time greater than or equal to 15 minutes. Again, rule of thumb is that one technician is performing and recording the agitation speed and start and end time of agitation, and another technician is verifying all these activities. Step number 10 is obviously temperature. This step is telling the technician to keep the temperature of the reactor between 75 to 85 degrees Celsius. Moreover, the time duration is provided that this temperature range should stay within the specified limit, on for about 180 minutes or more, which means after 180 minutes of mixing, the desired chemical has formed. The temperature range is also very critical. As you can see on your screen, step 10, the yellow underlined step instructions that if the temperature range is out of the specified limit, you must inform the supervisor right away. Now, just in case, if the temperature falls out of the limit, do as instructed and call the supervisor and absolutely nothing else. At this step number 10, the 180 minutes is the reaction time. The scientists have established that after 180 minutes, the chemical reaction has taken place and we should have the desired new chemical by now. Therefore, the instructions on step number 11 is telling you to take the sample at this time. If you remember, on the third page of the BPR, the instruction was given to take the samples only from the sampling valve that is attached to the reactor. The second sub-step in step number 11 is then asking you to record the time when sample was taken. Further, it is instructing you to send the sample to QC lab. At the time of sampling, you need to fill a log with initial and date. Do you remember? It is FDA compliance part, and it is necessary for traceability purposes. Therefore, the sample number and lot name and number is recorded in the log. Then this step talks about if the sample results are pass, then proceed to the step 12, which means this test confirms that the reaction has taken place and we have the new chemical composition ready. It also talks about if the test result is not pass, repeat the step 10, which means you will have to do the process of the agitation time and temperature, the whole reactor process again, since the reaction hasn't happened. But this is just in case scenario, this is validated procedure. So 99.99% of the time, there is no need to do the reaction again, since it works as intended every time consistently, since it is a validated procedure. Step 12 is now asking the technician to cool the content of the bioreactor between 20 to 25 degrees. This temperature range can be adjusted and achieved with regulator since we now have achieved the desired test results. Technician shall record the exact temperature and verifier shall verify the activity with initial and date. In this step 13, under sub-step 1, we are ensuring that the temperature shall stay in the range of 20 to 25 degrees, and at the same time, sub-step 2 is instructing to apply vacuum so that the excess solvent is removed from the bioreactor, so that we achieve the target volume of the reaction mixture. Importance of this step is that slurry will be thick and will be easier to process in the next step. Technician will record the temperature of the bioreactor, vacuum start time and its reading. Verifier will verify all the activities with initial and date. Step 14 talks about to take a 10 ml sample from sampling valve at the same time. The The sub-step is asking technician to measure the pH of the slurry. Slurry is nothing but mixture of insoluble matter suspended into the solution. A slurry is a mixture of denser solids suspended in liquid, usually water. And step instructs that pH should be less than or equal to 4. The technician shall record the actual reading in verifier, verify the correctness of the activity. Step 15 is talking about a filter dryer. What is this filter dryer? This is kind of an equipment which is used to separate the solids from the mixture. In this step, technician will be ensure the cleanliness of filter dryer, visually also update the logbook before transferring the slurry into the filter dryer. Verifier also verifies the filter dryer status to ensure only clean equipment is being used. This is a GMP practice. In step number 16, manufacturing technician checks the leakage in the filter dryer by applying pressure, which will be 2 to 4 bar. Also, verifier will verify the activity. Now we need to push this slurry into the filter dryer, and in order to do that, we need to set a pressure in the bioreactor, as you can see in image number 1, which also can be controlled by a pressure regulator. The slurry will be transferred with the help of transfer pump into the filter dryer. The transfer pump actually speeds up the transferring process. This transfer pump looks like image number 2. Under the sub-step of step number 17, after collecting slurry into the filter dryer, manufacturing technician will apply 1 bar nitrogen pressure into the filter dryer, as you can see picture number 3. Now in the filter dryer, spin the slurry at the 150 RPM. This will separate the solids from the mixture. For an example, the function of filter dryer as a washing machine spin tub. When spin tub rotate at high speed, it separates the water from the cloths. After completion of filter drying process, we get the solid cake as a drug substance or API. As you can see cake in the picture number 4 in white color. Manufacturing technician will record all these process parameters and their readings in respective sections, and verifier will verify all activities with initial and date. In step number 18, we need to wash the cake with ethanol. This process repeat until washed liquid appears colorless and transparent. This process will remove all the unwanted impurities from the cake. Cake is nothing but our API in raw form. Manufacturing technician will perform this activity and verify will record with date and sign. After removing all the impurities, our cake contains some moisture. Then we need to dry this cake. Step number 19 talks about to dry this cake with nitrogen and apply 1 bar pressure for more than 30 minutes. This process performed in the filter dryer. After drying, this cake will not convert in solid mass, it will remain in amorphous form or in powder form. Before packing of this powder in bag, you need to check weighing balance, visually cleaned, and last calibration date done. Also, you need to perform weighing procedure as per instructed. And all these mentioned in the BPR section as instructed in step number 20. In the step number 21, after completion of drying process and weighing balance status, you need to fill the DS in appropriately sized polythene bag, weigh, and record the weight here clearly as per GDP. As you can see on your screen, gross weight, tare weight, and net weight, all these fills this performed by section, and verifier will verify this step. It is an important part, make sure correctness of all entries. Step 22 is instructing to affix the label provided to the polybag in which the DS is packed. This label is provided along with the BPR by the documentation department. Furthermore, step 23 provides the range of DF001, which is equipment number deep freezer, and the range is -10 to -20 degrees Celsius. First sub-step is instructing technicians to place the drug substance in deep freezer. Sub-step 2 is instructing you to record the temperature at the time of placement of the DS in freezer to ensure the right temperature conditions were followed. There is a note on the top for this step, which provides the range of -10 to -20 degrees Celsius. Let's understand why this range. Most DS require below freezing temperature, as they are usually not stable at above freezing to room temperature, something to remember. Now we are moving to the final part of the process of drug substance manufacturing. You have performed all the step-by-step instructions and have packed the final drug substance all labeled and ready to go for the process of drug product, which is DP. In the step number 24, final calculation is performed. You will see this practice across the board globally. This is a formula that is used to estimate if any extraordinary losses occurred in final DS quantity due to maybe an error in process somewhere. So first of all, let's understand the theoretical yield. This calculation is nothing but an estimated amount established by the process engineers and scientists. This is an estimated amount that they believe that you should achieve if all manufacturing process is followed properly. Theoretical yield is given in this batch at the beginning, and the actual yield is the amount of drug that you have manufactured and finally achieved after following step-by-step instructions in BPR. So now you will take the actual yield that you have and divide it by the theoretical yield and then multiply it by 100. Once the percentage of the yield is calculated, you will record the calculated percentage here, and again, another technician will verify at verifier's space in the BPR. Verifier will also check these calculations as well for accuracy. Now look at this page. This is usually one of the last few pages of the BPR. You have learned about deviations in terminology class as well. Again, for your reminder, anything that doesn't go as intended during the manufacturing process is a deviation that must be recorded here with its brief description and conveyed to the supervisor as well. That is all. The very bottom section is for the history of the BPR. Anytime if there is an update to the BPR, even if a spelling mistake correction from when the BPR was created first. Every time something is changed, another version will be created, and the reason for the change will be mentioned here in this BPR. This is part of traceability, which is required by the FDA. We have discussed the traceability part over and over again. Now you have successfully completed your first assignment and know the DS process with use of bioreactors. If you get a chance later to work for a company that manufactures DS only, you know right off the bat that you will be working with a process similar to this. The drug recipe can be different, but the bioreactors, their temperature, agitation speed, timing, in-process samples, gowning practices, buffer preparation, pH readings, sample submissions and logs, your understanding of who will issue the BPR, where you would go next to receive the materials, who would sign which log is the information you need to understand and learn to become a great technician. As long as you have understood the GDP and GMP principles, understand the importance of filling logs, creating full traceability, following training compliance, not executing on anything that you are not trained on, make sure to make a practice to always check calibration and cleaning tags of equipments before using them for drug safety. These are the very common mistakes that happen day in and day out in our industry. If you know and understand and make these practices a part of your professional profile, then you are already ahead of the game in your profession and a valuable technician that a company would like to keep. See you in the next training, where we will take this DS and will turn that into a drug product, yes, DP. See you there.